Hi everybody. So today I wanted to talk to you about what normal cell division looks like. I posted some slides and I asked you to do some review vocabulary, and this is slide 20 in the cell cycle slides that I posted I think on March 24th, and I promised that the classroom will be much more organized now that we're up and running. I was just trying to get things into your hands so that you could do some work. So today we're pretty much going to review mitosis. I'm going to do short videos that you can work through. I think it'll be easier for me to upload and for you to actually stream, especially if you're out in the country and have spotty internet. So I didn't want to do really long ones that you have to try to get open. So if you think back to regular bio, we talked about how cells make new cells. When we talk about mitosis, we're talking about how we make toes and fingers and eyes, how our hair grows. Speaking of hair growing, I have roots this long right now. They're great. I hit them today. So this life cycle that a cell goes through, it starts here, right, as one, and we're going to end up with two identical new cells. 90% of this cell cycle is spent in interphase, and 10% is spent in mitosis, where it actually divides. And the most important part of this is interphase. Interphase has three parts: G1, S, and G2. And this is so important because this is when the cell grows, and this is when the cell doubles everything. So that when it gets to mitosis, it can make two new identical cells. So if you think back to biology, we talked about IP mat. I was interphase, P prophase, metaphase, anaphase, telophase. In AP Bio, sometimes pro-metaphase comes up, but it's not important at all. So it's right before metaphase starts. So let's look through the steps. You're going to remember these, or hopefully they'll start coming back to you. And if you're a pack rat, you might still have your bio notes. So I'm going to go back to this really quick. Sorry, I'm talking to no one. This sucks. It's really awkward teaching like this. Interphase has three phases. And I'm going to go way, way back. This is at the very beginning of all of this. Interphase has three parts. G1, where the cell grows, and it makes more cell membrane, makes tons of cytoplasm. S, which you should have defined, is when there's more growth, but more importantly, the chromosomes, or all the DNA, is doubling. Then G2, which is still part of interphase, is more cell growth and all the organelles double. Sorry, not divide. So the important part of this is that we understand that interphase, 90% of the time, the cell is preparing to split. So it has to double everything, not just its DNA. It has to have enough mitochondria. It has to have enough chloroplasts if it's a plant cell. It needs to have enough lysosomes. It needs to have enough endoplasmic reticulum. So everything in the cell doubles during interphase. Interphase has the three parts, the two growth phases, and then the S phase. And then we go into mitosis, which I'm going to skip back to where I just left off. This is a great video on growing skin because we understand the cell cycle, so don't skip it. And then I'm going to skip over controlling the cell cycle today, and we'll go back to tumors later this week. So, mitosis, what does it look like? So the first thing that happens after interphase, where everything gets doubled, is prophase. And that is when our chromosomes pair up. So you will always see them in these pairs. This is one pair. This is two chromosomes. Chromosomes like to stick together. The nucleus falls apart and the centrioles become visible. These are these things that are going to allow the spindle fiber to form. Metaphase is when they move to the middle, and they align on what we like to call either the metaphase plate or the equator. So P for prophase is they pair up. M, metaphase, they move to the middle. They're now on the equator, these perfect little pairs. Dude, metaphase starts in 5 minutes. I can't believe you're not condensed yet. So think about chromatin versus chromosomes. So here's that pair of chromosomes. It's condensed chromatin. It's all just DNA. That's it. Anaphase is when they pull apart, right? A is for apart. So the sister chromatids, those pairs get split. And so now we have chromosomes heading to each pole of the cell. The cell starts to get long. And then finally in telophase, we have two new nuclei, and we talked about telophase being towards the poles, right? So they're towards the poles, completely apart. We see the nucleus coming back, and the cytoplasm is being cut, and we call this cytokinesis. This is a cool picture of cytokinesis happening in an animal cell where you can actually see it. So I would suggest today that you watch this video and this video, and thank you, Savannah, for reminding me that I had unlocked all of these. So now they're all unlocked. You can watch them. Review the steps of mitosis, and tomorrow I will post all of the stuff about the control of all of this and why sometimes this goes wrong and tumors can form. So take time to watch this, think about it, watch the videos, go through the notes or the review vocab that you have already done, and please respond with a comment in Google Classroom so that I know that you're doing this. This is up to you. You need to be college ready. See ya.